Hello, my name is Anaya Sharma, and this is my presentation on the article Amygdala and Hippocampal Volumes in Adolescents and Adults with Bipolar Disorder. The reason I chose to write about this article is because I have always been interested in the disorders of the brain. I have experienced anxiety disorders myself, and although they may be different to bipolar disorder, I can attest to the fact that these disorders really alter the human brain. Specifically regarding this article, it talks about how the researchers want to figure out whether the hippocampus and amygdala decrease in size due to bipolar disorder. I think this is a super interesting topic because it may show how mental disorders can have a physical impact on our brain. The topic of the hippocampus and amygdala relate to our studies in class because we learned about the limbic system, which helps regulate our emotions. Because the hippocampus and amygdala are a part of the limbic system, it makes sense why there may be a decrease in volume for these parts due to bipolar disorder, because the regulation of emotions when having bipolar disorder is a little more challenging. The research question that this article aims to answer is, does bipolar disorder cause the amygdala and hippocampus to decrease in volume? This research question can then be turned into the hypothesis stated on the slide. Due to the effects of bipolar disorder on the brain, the amygdala and hippocampus decrease in size. This article aims to study the relationship of the limbic system and bipolar disorder and how to treat bipolar disorder more efficiently. Operational definitions are descriptions of important words in terms of a procedure, action, or process that will be undertaken during the research. Some operational definitions in this article include bipolar disorder, amygdala, and hippocampus. Bipolar disorder in this article is defined by clinical interviews that were carried out by board-certified adult psychiatrists who tested for the presence of bipolar disorder. With the amygdala and hippocampus, these were defined by means of tracing, which means they used imaging to scan the brains and then trace the outline of the hippocampus and amygdala. This helps correctly identify the size of the two parts of the brain. The last bullet point is something I want to go into deeper in another slide, but it talks about how the images were taken and what procedure was in place to make sure that everyone's scans are taken the exact same, so there is no variation and margin for error. Some important concepts that are talked about in this article are rapid cycling, morphometric analysis, and axis 1 disorders. Rapid cycling is a pattern of frequent and specific episodes that happens in people with bipolar disorder. This is specifically 4 or more episodes of mania or depressive episodes in a 12-month period. Morphometric analysis is a concept that analyzes both the size and shape of an organ. This helps to gauge the size of the organ and make sure that researchers are able to get accurate conclusions on whether the hippocampus and amygdala are actually smaller than a control brain. Lastly, an axis 1 disorder is one of 5 axes that encapsulates clinical disorders. Bipolar disorder is classified as an axis 1 disorder. In total, there were 36 participants with bipolar disorder and 56 participants that were control subjects. Out of the people with bipolar disorder, there were 14 adolescents and 22 adults. With the bipolar disorder group, the way they were chosen is that they were outpatients at specific hospitals in Connecticut. 12 out of the bipolar group were female, and 8 were free of medication. In the adolescent group, there were 8 females and 6 were off of medication. The bipolar patients range from age 10 to 54, ages 23 to 54 in adults, and 10 to 22 in adolescents. For the control group, there were 23 adolescents and 33 adults. For the healthy control group, they were recruited by word of mouth and advertisement around the community. They were then selected randomly from a list of 10,000 potential subjects living in the same areas as the people with bipolar disorder to account for similar socioeconomic backgrounds. Approximately 10% of the eligible controls contacted were decided to participate in the end. There were 25 adolescents and 33 adults, aged 10 to 57, ages 24 and 57 in adults, and 10 to 22 in adolescents. In the control group, there were 17 females in adults and 10 in adolescents. I believe this group is a great representative sample because there are a similar amount of males and females, and it covers a wide age range. It also takes into account socioeconomic backgrounds. The design for this study focused on the brain scans of bipolar patients and control subjects. The researchers needed to take into account clinical variables like substance abuse and medication that could alter the brain. To start the research, clinical interviews were conducted by psychiatrists to check for presence of bipolar disorder. The independent variables are rapid cycling, medication, and substance abuse coupled with bipolar disorder. The dependent variable is the volume of the amygdala and hippocampus. The way this study started was that the researchers took scans of the hippocampus and amygdala to check their volumes. This was done on a single 1.5 T scanner. Head positions were standardized during the scans so that there is no variation in imaging from person to person. These scans were then analyzed using repeated measures statistical analysis to compare the volumes of the hippocampus and amygdala across all patient scans. Age and sex were factored in this analysis. Subjects were not allowed to drink or use street drugs 24 hours prior to the scan to make sure all data they collect is accurate and not just an effect of these substances. To get the most accurate results, researchers used tracing to outline the two parts of the brain. Differences in spatial agreement, or the amount of space occupied, was corrected by an expert. The results that were documented showed that the amygdala had a significant decrease in the bipolar patients, and that the decrease was 15.6% smaller than the control group. However, for the hippocampus, it only decreased 5.3%, which wasn't statistically significant. According to reports, there was also no significant effects of rapid cycling, age, medications, or substance abuse that would be able to explain the significant decrease in volume loss for the amygdala. The graph shows the volume differences between the bipolar group and the control group for both the amygdala and hippocampus. The major conclusions drawn from this research was that bipolar disorder has evidence of structural abnormalities. This means that bipolar disorder has more than just mental effects, it has physical effects as well. Another conclusion is that the decrease in volume of the amygdala and hippocampus could represent a genetic vulnerability to bipolar disorder or earlier effects of the disorder. This makes sense based on the research gathered because the patients with bipolar disorder had already had this disorder for a long time, so this decrease in volume would have happened much earlier on in their disorder. To identify if this is correct, another study needs to be performed where they look at the amygdala and hippocampus of someone who has just been diagnosed with bipolar disorder, and then rescan a few years later to check for any significant decreases in volume for these two brain parts. This would be a longitudinal study. Another study that could be performed is checking the brains of direct family members of patients with bipolar disorder and seeing if the decreased amygdala and hippocampus are genetically linked with a predisposition to bipolar disorder. The conclusions drawn by this study are also valid because it corroborates the results of many other studies that have reports of decreased hippocampal and amygdala volumes due to bipolar disorder. The practical implications of the study are restricted due to the limited ability to generalize these conclusions. This is due to the fact that the probability of the different subtypes of the disorder and the course of the disorder varying between patients. A longitudinal study, like the one I mentioned in the previous slide, would be able to more accurately define the structure of the amygdala and hippocampus at the beginning, middle of treatment, and after a few years of treatment. Studying prepubescent samples along with these longitudinal studies and family members of bipolar patients can help identify the phenotypes associated with the disorder. Lastly, this study can help clarify the developmental aspects of bipolar disorder. This could help society figure out how the limbic system is altered due to bipolar disorder, and how the disorder progresses over time. It makes an impact on the physical rather than just the mental, and this changes the whole stigma around bipolar disorder. It is not just a mental illness. It has real physical effects that can cause damage to the brain. This is a complete APA reference for the study I talked about. Thank you so much for watching my presentation.